immunity and infection, biochemical approaches to therapies. Today, on 18th June 2020, we have two sessions scheduled. The first session is scheduled at 11 to 12 p.m. and the speaker of the first session is Dr. Puneeth Kumar, the founding director, Azymus Life Sciences Private Limited, and he is going to speak on genetic testing in immune disorders. And the second session is from 12 to 1 p.m. and the speaker of the second session is Dr. Poornima Priyadarshini, and she's a scientist at CSIR-CFTRI, and she's going to speak on food bioactive as immunomodulators during viral infections. Now, it's my pleasure to invite one of our faculty member, Dr. Poornima Madam. She is an assistant professor at Department of Biochemistry, Davangere University, and I request Poornima Madam to introduce the first session speaker, Dr. Puneeth Kumar. Poornima Madam, please.
Thank you, Pramod sir. Good morning, everybody. It is my immense pleasure to introduce our today's guest speaker, Dr. Puneeth Kumar, Founder Director, Azymus Life Sciences Private Limited, Chamarajanagar. Dr. Puneeth Kumar did his MSc in Biochemistry from University of Mysuru in 2002 and completed doctoral degree in 2009 in CFTRI, Mysuru. Then he moved to Stanford University, Stanford, USA in 2010 for his postdoctoral research. Then sir worked as faculty scientist in Institute of Bioinformatics, ICPL Bangalore, and also served as guest faculty in the Department of Molecular Biology, University of Bangalore from 2013 to 2015. Then from 2015 to 17, sir has worked as assistant professor in PBMM PG Center, Mysuru. Then sir also has working experience as scientific affairs manager in European Clinical Genetics India and MedGenome Labs Limited, Bangalore from 2007 to 2019. And sir guided nearly 7 students for their project work for MSc degree and has 7 research publications in reputed national and international journals, and also nearly 13 abstract or paper presentations in various conferences and symposiums. Awards and honors of Dr. Puneeth Kumar include Dean's Award from Stanford University, USA, Travel Support Award from American Society of Nephrology, Philadelphia, Postdoc Fellowship from Stanford University, USA, Senior Research Fellowship from ICMR, New Delhi, India, and Project Research Fellowship from Department of Atomic Energy, DAE, Mumbai, and DST, New Delhi, India. And Dr. Puneeth Kumar has professional memberships in American Society of Nephrology, ASN, USA, and Society of Biological Chemists, Mysuru chapter, and 4-hydroxynonenal HNE Club, Graz, Austria. Today, sir is going to speak on genetic testing in immune disorders. With this very brief introduction, now I request Dr. Puneeth Kumar to start his session. Over to Dr. Puneeth Kumar. Thank you.
Yeah. Dr. Puneeth Kumar, please share your slides and you can go on your presentations.
Thank you, Dr. Prabodh and Poornima for giving a nice introduction. So I will start my session right now. Pramod, so please give me the sharing option, Pramod.
Yeah, I think the share option is given for you. You just go to the share, click on the share.
No, host disabled the participant for screen sharing, it's giving.
Okay, I will ask. Pratap?
Yes, sir, yes, sir.
Yeah, please give the share option. Is enabled? Oh, Dr. Puneeth sir, sorry, I mean, there is some, just 1 or 2 minutes, so we'll give the option for sharing.
Received. No, it's shared.
Yeah.
Is it okay?
Yeah. Yes. So you can make it full slide. You can just make the full slide.
Okay. So hello everyone. Today I'll be speaking on the implication of genetic testing in autoimmune disorders. Hello, can everyone hear me? Am I audible?
Yeah, it's very clear.
Okay.
Yeah, yes, the voice is clear, everything is clear. You can go ahead with your presentation.
Okay, thank you. So my talk will be having two parts. So in the part one, I will be talking on I'll be giving an overview on what is the genetic testing means and what are the techniques that are available in the genetic testing. And in the part two, I will give an example or I will explain how this genetics testing can be applicable, taking an example of a case study and how these results can be analyzed and interpreted so that it will be used for the clinical settings. So the identification of the genetic testing, how it can be applicable for the clinical side can be discussed in a part two. So let me start with part one, where I will be giving an overview of the genetic testing. So we all know that autoimmune disorders arise when our immune system fails to recognize or distinguish between the self and the non-self constituents in our body, which can lead to a large-scale inflammation and tissue damage. And though the most of the autoimmune disorders, the cause for the most of the autoimmune disorders are unknown, so several studies have clearly said that there is a genetic predisposition that is responsible for the development of autoimmune disorders in many of the cases. Today, American Autoimmune Related Disease Association, so they have listed more than 100 diseases as a autoimmune disorders based on the involvement of the immune systems for the development of these particular disorders. So that is American Autoimmune Related Disease Association, they have categorized it. So here we can see some of the autoimmune disorders that are in one of them, like rheumatoid arthritis, you can see. So you can see the inflammation and dermatological synapses that can be observed due to immune response or inflammation that is caused due to overactivated immune cells. So coming to the central dogma of this molecular biology or life, what you call, so here the functional protein that is being synthesized in our system is being programmed by gene. So gene undergoes different stages like transcription, splicing and translation for giving a gene product that is a functional gene product, which you also call it as a protein. So any abnormality or any change in the sequence of this gene, which we can call it as a program directory, can lead to a formation or the production of a abnormal protein or a non-functional protein. Here abnormal protein means here the protein will not be completely synthesized. It will be like 70 to 60% will be synthesized due to mutation in certain genomic regions. Okay, so where it can be partially functional, that is abnormally functional, or it may be overactive functionally, it may be overactive. In other words, it may also lead to these mutations or the variation in the sequences in the gene can also lead to a non-functional protein. So that means these proteins are of no use, or they may not be functional and they are not responsible to carry out any of the cellular activities. So when you look into the roadmap of these, the genetic testing and the implication in identifying the genetic disorders, you can see in the early 80s, so the studies were due to the limitation in the availability of the technology. So the studies were done based methods, and the studies showed that the one gene is responsible for one phenotype. That means if you observe a particular genetic disorder, for example, sickle cell anemia or phenylketonuria or Fabry diseases, so they found that there is one gene is responsible or behind as a causative factor for one phenotype that has been observed in the subjects. Later, the studies increased or the advancement in the technology showed that one gene can also be responsible for more than one phenotype. So that is, here I have given an example like RET gene, which can be responsible for ischemic disease and also multiple endocrine neoplasia. And similarly, RYR gene was found to be responsible for or can implicate in the development of malignant hyperthermia and also central core disease. So like that, as the studies increased and the advancement in the technology increased and the sensitivity of the detection of the sequencing technology increased, so finally we found that many genes can also implicate in one phenotype, or many genes can become a causative factor for developing a one phenotypes. For example, here you can see a more than 300 genes were found to be causative or or applicable, or they have the implication in development of the intellectual disability, and more than 100 genes were found to be responsible for development of epilepsy and also neuromuscular disorders. So likewise, as the technology and the research developed or advanced, so more and
Hello? Hello? Hello, Puneeth? Hello, Puneeth?
hello.
Hello? Yeah, now, so
Okay.
there was, yeah, you can share your slide.
Okay, it's sharing now.
No. I think now your picture is visible. You have to reshare it. There was internet disconnect, I think.
Okay.
You can reshare your slide. Yeah.
Fine.
Yeah, fine. Go ahead.
Okay. So I was discussing about the monogenic and the complex genetic disorders. So where the monogenic or a single gene disorders are rare, and if any variant or a mutation that occurs in those gene can definitely have a high risk of developing the genetic disorders or a particular disorders. So these are, though they are rare, but any mutation or a pathogenic variant that is observed in that gene can be have a high predictive risk or a inherited risk of developing the genetic disorder. So the other part of the genetic disorders is where you call it as a common complex genetic disorders, where it is a common and a number of genes are involved here. Along with the number of genes, multiple genes, even the environmental factors do play an important role for developing the potential disorder. So that means they have they are not too many risk in complex disorders, because they are common and more than one genes are involved in developing the disorder, and they are having an empirical risk, and the chances of developing the subjects may be a carrier, and they may not show the disorder or the phenotypes that is responsible for the that is due to the mutation that occurs in any one of the genes. So the mutation should occur in all the multiple genes that are involved, and also along with that, even the environmental factors should be supporting to develop a subject for that particular phenotype. So what are the different types of mutations that are that we observe in a gene, so that can lead to a disorders? So you can see a substitution in the gene sequence, that is the base pairs, that is A to G. Here you can see, and you can also observe the insertion, that is the additional base pairs will be added to the whatever the normal sequence that observed in the normal gene. And there is also, you can observe the deletion of a particular base pairs. Apart from this, you can also observe a large copy number variations, where a large part of the segment of a gene can be changed. There may be, there may be there may be variation in the large part of the sequence itself. So that is called as a large copy number variations can also be observed, And these are the different types of mutations that can be observed, and they can result to the disorders. And what are the, and coming to the classification of these mutations, it may be either heterozygous or the homozygous mutations. So we all know that each gene, there will be 2 alleles for a particular gene in a chromosomes. So if these mutations occur in any one allele, and mutation in that one allele can result in a development of the disorder, so that is called as a heterozygous mutation. If the mutation has to be occur in both the alleles, and then and the mutation in both the alleles will result in a development of the particular phenotype, means, so that is called as a, that you can categorize as a homozygous mutations. So these heterozygous and homozygous mutations, heterozygous mutation can be autosomal dominant manner, it can be inherited in an autosomal dominant fashion, whereas homozygous mutations can be inherited in an autosomal recessive fashion. So these are the different types of mutations that you can observe, and you always note, you always screen for these kind of mutations when you do a genetic testing. Then coming to different ways to correlate, what are the different approaches to correlate these mutations that occur in the gene to the phenotype or the whatever the subject? So you have 2 different approaches. First is a genotype to phenotype correlation. So that means you analyze the sequence of a person's DNA. So that means entire DNA will be sequenced, and you will identify the mutation which is present. And now after identifying the mutation, and now you correlate that mutation which explains
Hello, Puneeth. Hello, Puneeth sir. Puneeth sir, may I audible? Hello, Puneeth sir. Please, I request all of you to be little patient because he's going to reshare it. There was some network problem with him. Hello, Puneeth sir.
hello.
Yeah, it's clear. You can go ahead.
So the second approach is what I was discussing is the phenotype to genotype correlation. So where first you identify the phenotype in a patient, and you mark it whether it is to a particular organ. That is means whether the phenotype that is being observed is particularly to central nervous system, or cardiac vascular system is being affected, or ophthalmological system is affected, or immune system is affected, or renal system is affected. So you first categorize the which area of the system is being affected, and based on that we already have a database that have been listed the number of genes that are responsible for various disorders of that particular organ or a system. So that, for example, if you take a renal system, you have a particular genes that have been already available in the database. So that explains why this renal disorder has been occurred. So similarly, even if you consider the immune system, so there are certain genes that have been already available and listed by OMIM, that is Online Mendelian Inheritance of Man. So in this website, there are genes that are available, so which are responsible for immune related disorder. So when you identify a subject which is being clinically showing some immunological disorders, means then you can directly screen those immune related genes. So no need to screen the entire gene, entire DNA or entire genome or the whole exome. So it's only those particular genes can be screened, and you can directly target the responsible gene and then go for a treatment modalities. It helps the clinician to decide the treatment modalities. So this is the other way of genotype to, correlating the genetic testing to a phenotype. So that means the phenotype to genotype correlation. So then how these genetic testings are done, then what are the techniques that are available for this genetic testing? You know that in human genome, there are 3 billion base pairs are available. So the human genome consists of around 3 billion base pairs. Of it, around 99.6% you can see the identity, inter-individual identity can be seen. Even if 99.6 inter-individual identity is been found, yet there are 4 million differences can be observed in the sequences. So the human genome consists of 3 billion base pairs, and even in the 3 billion base pairs, around 4 million different sequence differences can be observed from individual to individual. So you all know that a gene consists of a coding and a non-coding regions, that is the exons and the introns. So totally we have around till date online Mendelian inheritance of man. So they have listed around 24,600 genes have been listed in them, and out around 23,000 genes are the coding regions. So that means there are 23,000 genes, so they can give for a particular protein product or a gene product. So of the 23,000 genes, they have around 8,000 genes have been listed from OMIAN as a clinical exome. So that means these 8,000 genes are responsible for, are being listed as the responsible or the causative genes for whatever the genetic disorder that have been listed till today. Till today, whatever the genetic disorders are available in the website or listed, so they are being categorized, or all these are caused by around 8,000 genes. So that means 80%, 85% of all the disease causing mutations are positioned within the coding exon region. So that means if you take the 3.3 GB as a 3 billion base pairs, so around approximately 1% of it will be the coding regions, and around 0.5 MB will be your targeted exome. That means around the clinically relevant genes that are responsible for the disorders that have been genetic disorders that have been listed till date. Then how then genetic testings are done for what different reasons? So that is other questions we have. So genetic testings can be done for diagnostic testing. That means if you have a symptoms of a disease, and secondly it can be used for pre-symptomatic and predictive testings. That means in a family there is a history of a genetic condition, so that you can do the genetic testing in those family members, so that the offsprings or their family relatives, to predict whether they have the risk of getting these genetic disorders can be predicted by doing this genetic testing. And you can also do a carrier testing, where the family members there is a genetic history of in a family, genetic disorder history in a family or a ethnic. Hello, can you hear me? Hello, hello.
Yes, sir. Now you are audible. Please, you can continue.
Okay. Okay. So this genetic testing can also be applicable for pharmacogenetics. So that means to determine the intolerance or the dose response in a particular subject who is undergoing a treatment. So that I will explain this with an example in a later slide. So how this genetic testing can be is implicated for this pharmacogenetics. So the genetic testing can also be applied for knowing the prenatal testing. That is, you can know whether the baby is at the risk of developing the Down syndrome or trisomy 18. So these are these kind of genetic disorders can also be predicted by using a prenatal genetic testing test. And also you can do a newborn screening where the babies that have been newborn babies can also be predicted for the risk of developing phenylketonuria, congenital hypothyroidism, or any other neurological disorders can also be predicted by using the genetic testing. And finally, even the pre-implantation testing. So that means whether the embryo or the semen that is being used for IVF or IUF treatments can also be checked using the genetic testing for whether they are the carriers for any of the genetic disorders, or they are having any high risk of developing the genetic disorders if the embryo has been implanted and developed. So what kind of samples are used or expected for the genetic testing? So you can use the blood samples, and you can use the buccal swabs, or you can use the amniotic fluid or chorionic villus sampling for any prenatal genetic testing, and also for any tumor issue to identify the genetic factor for development of the tumors. You can use the FFE blocks, that is paraffin blocks of a particular tissue. So coming to the techniques that are available for the genetic testing. So you have a molecular cytogenetic testings, and also we have a advanced sequencing technology to identify a mutation in a gene and its correlation to the observed phenotype. So molecular cytogenetic testings includes the karyotype and the karyotyping, and also the fluorescence in situ hybridization. Whereas the advanced sequencing technology includes Sanger sequencing, next generation sequencing, multiple ligation dependent probe amplification, which you call it as MLPA, and another one is a genomic microarrays. So these are the different advanced sequencing technologies that can be used for identifying the genetic disorders. So we all know you already know that what is karyotyping. So karyotyping is done based on the G band banding pattern, and to determine whether there is a portion of the chromosome has been deleted or inserted, or the entire chromosome itself has been added. There is a duplication of the entire chromosome can be detected by using the karyotype. This is the very primitive stage or a primary type of screening that is done in most of the clinics to identify a genetic disorder. For example, here you can see a baby having a Down syndrome. So where you can see there is an extra chromosome in the chromosome 21. You can see there is a extra chromosome 21, so which leads to Down syndrome in this baby. So this kind of analysis or detection can be done by using the karyotype. So that means if any micro deletions cannot be detected, the limitation of this karyotyping is the micro deletions cannot be detected here. So to detect these micro deletions, that means the deletions at the gene level itself. So that means a particular gene or a particular segment of a gene is being deleted means. So that can be detected by using the fluorescence in situ hybridization technology. This is called as a FISH. So where the fluorescent probes will be used to hybridize to the particular region of interest, and to check whether that particular region is being deleted or it is being added. So there may be chances of deletion or addition of a particular gene. So that is called a micro deletion or micro addition or micro insertion. So that can be detected using fluorescence in situ hybridization. Pardon? So here I can show you an example here. So this
Hello, please switch off your microphones, because we are getting some background noises and it's going to disrupt the audibility of the speaker. Please turn off your microphone. Sorry, Gouda PA, please switch off your microphone.
Hello.
Hello, yeah, Pune, it's clear. Puneeth sir, it's clear. You can go ahead.
Hello. Yeah. Okay. So here, taking an subject where he is suffering from death muscular syndrome. This is called as a DMD. So here DMD is caused by a deletion of a exon region either 1 to 51. So exon from 1 to 51 will be deleted from a subject in a gene. So which results in a abnormal protein. So that means the protein will not be completely synthesized. It around 60 to 70% of the protein will be synthesized and rest of the protein will not be synthesized for to make that protein a functional protein. So that is so to determine whether that which exon region is being deleted, whether it is from exon 1 to 51 or particularly some 25 to 51. So this can be detected using the FISH, that is fluorescence in situ hybridization, using the probes that are particular for that particular sequence. So here you can see here in a normal control, you can see there are two signals are there. So one is the green signal is a normal control and pink signal is the chromosome region of your interest. So here in a healthy individual, you can see there are two signals that are available, that is two green signal and two pink signals. So that means you're having the entire exome region is being present in that chromosome. Okay, so when you look into the patient with the deletion, so here I am seeing a DMD patient sample is been taken. So here you are suspecting there is a deletion in that particular exome. For example, it is in exome 25 is been suspected to be deleted. So you have a probe or a sequence which is particularly for that exon region, and you try to hybridize it. Okay, in a normal C, 2 plus 1. So that means there are two green signals, but there is only one pink signal is there. So that is one of the gene of your interest or the region of the gene is missing in this patient. So which is relating to the which is responsible for the development of this DMD, that is death muscular dystrophy. So this is how the FISH or the fluorescence in situ hybridization gives you an idea or the data. So the saying that this is the reason or the deletion of this particular exon region is responsible for whatever the phenotype that you are observing in this subject. But the limitation is this FISH cannot identify the single gene mutation. I mean single mutations or the single insertions or deletions cannot be detected by this FISH. That means which region has been mutated, whether it is A to G or G to C or what kind of mutations cannot be detected here, detected in the using this technology FISH, that is fluorescence in situ hybridization. So now to do this, to identify the sequence variation, so now we have to use advanced sequencing technology, that is from Sanger sequencing to next generation sequencing technology. Sanger sequencing technology is till today it has been referred as a gold standard technology, because here you will be using a fluorescent labeled modified nucleic acids. So that means dideoxy ATP nucleic acids will be or base pairs will be used for detecting the mutation. But the limitation of Sanger sequencing here is you cannot screen a larger kilo base pair deletion, mutations. So that means you can screen in one single sequencing, you can screen one exon region or two exon regions at the maximum. So that there is a limitation. But when you look into the entire genome, you have a 3 billion base pairs. So you can imagine, so the how the time required for screening the entire 3 billion base pairs means it may take like decades. Using the Sanger sequencing, it will take decades to screen the entire 3 billion base pairs or the entire genome. So that is the limitation here. But when you come to the accuracy and the specificity of the test, it is still recognized as a gold standard technology. So coming to the most robust and sensitive technology till today, it has been used in various genetic sequencing labs, is the next generation sequencing technology. So here, this technology is also called as a massive parallel sequencing technology. So next generation sequencing technology is called as a massive parallel sequencing technology, where your chromosomes will be randomly cleaved and it will be printed on your flow cell, and now your sample will be added to sequence the already attached chromosomes, or the sequence, or I mean the piece of chromosomes. That means parallelly in one single shot, you will be screening the entire sequence. I mean the entire DNA or the genome will be screened in a single sequencing run. So that means you have a piece of chromosomes. The chromosomes will be randomly cut, and they will be printed on the flow cell. And now you have the sequence available. So now using the polymerase, you do the regular sequencing, and you can identify the sequences that are present, or any mutations that are present in the gene sequence. So actually, to explain the next generation sequence, it requires one additional class, actually 1 hour class or 2 hour class is required to understand the exact how this next generation sequencing technology works. So here I am giving just an overview or a brief of this technology. So where around at a time you can sequence the same chromosome segment for 150 to 200 times. You can sequence it, so that you will not miss you will increase the specificity and the sensitivity of the detection of the mutation. So this is the advantage of the next generation sequencing. So you can sequence that one particular gene region to 100 to 200 times, whereas it is not possible in the Sanger sequencing. So that's why this next generation sequence can be applicable for a targeted sequence. That means you can target it for a particular gene of your interest or a particular gene region of your interest, or you can also target it for a particular exomes. So that means number of exomes. You want to screen for certain 100 to 200 exome region in a particular gene, means you can target only for that particular exome region, or you can also use it for a whole genome. Pardon? Hello? So you can use this next generation sequencing to screen the entire genome, including the exon and the intron. The whole genome can also be screened using by this single gene sequencing. So this is one of the most advanced technology currently being implemented in the genetic sequencing labs. So one of the limitation in next generation sequencing is you cannot identify the larger deletion and duplication. So that means there is a larger deletion in approximately around 100 to 200 kilobase per deletion is there. For example, in a chromosome means, or in a gene means, that cannot be detected by this next generation sequencing. Or if there is a larger insertion, the duplication of a gene means that cannot be detected in a next generation sequencing. So that can be detected by another technology, sequencing technology called as a multiplex ligation probe amplification, that is MLPA, which I am not discussing in this session. These two technologies, that is MLPA and the microarray, I am not discussing in this sequencing in this session. So when you, in one word, if you want to see the difference between the Sanger and NGS, you can see the Sanger sequencing, you can see you are targeting one gene at a time. So that means you are targeting one gene in a single run. So whereas next generation sequencing is you can capture the entire genome or the entire exome region or the coding regions in one single run. So this is the difference between the Sanger and the next generation sequencing technology. So, but after doing the genetic testing, so how do you classify these variants or the mutations that has been observed? So you can classify these mutations into pathogenic mutations or likely pathogenic mutations or variant of uncertain significance. So here, pathogenic mutations means there is this mutation that has been identified in the gene is responsible for is a sure shot. That means the mutation occurs means definitely that person or the subject will be in a high risk of developing that disorder. And most of the studies and publications will also have been supported that for that mutation, that this mutation is responsible for this genetic disorder. So if the gene mutation that we identify by using the next generation sequencing, if that mutation has been observed, means that mutation has been already reported to be a causative mutation for the particular disorder, means. So then you can classify that mutation as a pathogenic mutation. So in any patient, if you identify a mutation and it's been classified as a pathogenic, means, so it is clear sure that this is the reason, this is the genetic factor that is responsible for whatever the phenotype you are observing in this subject. And secondly, you have a likely pathogenic, means here the variant is very likely to contribute to the development of the disease. However, the scientific evidence is currently insufficient to prove this conclusively. So that means additional evidence is expected to confirm this assertion of the pathogenicity. So that means the mutation has been identified, and it is not completely you can reliable to say that this is responsible for this phenotype what is observed in this client or subject, but you need some scientific evidences or any other clinical synopsis support or clinical data supporting clinical data to say that this mutation is a pathogenic. So unless you don't have additional supporting evidences, you cannot classify that mutation as a pathogenic. Yes, they will be classified as a likely pathogenic mutations. And lastly, you have a variant, other classification called as a variant of uncertain significance. So this variant has been detected, but it is difficult to classify it as either pathogenic or a benign or a likely pathogenic because of the current available scientific evidence. So that means these scientific evidence that are currently available in the databases are not supportive to say that this mutation is directly responsible for whatever the genetic disorder that is being observed. So in those cases, the clinicians will recommend to classify this as a variant of uncertain significance mutation. So, but what shall the clinician do in case of this? If you get a genetic report as a VUS, that is variant of uncertain significance, means the clinician has to re-evaluate the phenotype, the subject, and do the additional testings, okay, in the clinic to see whether there is a additional data which can support for this mutation that is been observed, that has been already reported in the genetic testing. So this is about the part one, that is what I till now, the techniques that are available in the genetic testing. So now, how to implicate or how this genetic testing is applicable in a clinical side. So here I'm taking an example to make you understand how this genetic testing is applicable in a clinical side. So here we have a case where a 8-year-old male was represented, or he appeared the clinician with a fever, hepatosplenomegaly, anemia, purpuric rashes, jaundice, overproduction of T lymphocytes and macrophages of cytokines, and also particularly interferon gamma and tumor necrosis factor alpha. And also it was his lab analysis showed that there is activated T cells in the peripheral blood and reduced natural killer cell cytotoxicity, reduced T cell cytotoxicity. So these were the clinical synopsis or the clinical phenotype or the data that was observed in the clinician in this subject. So now, so when you look into the clinical phenotype, so it is sure that it is a multi-organ disorder. So that means here you have a liver affected, spleen affected, even the brain is also affected, and there is a increase in the T lymphocytes and macrophages. So that means, so these data implicate that this may be a, or this can be a, this subject is affected with a familial hemophagocytic lymphohistiocytosis. So this is hello. 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 Hello.
Sir, please continue, sir. That is
Okay.
from other
So because I had a
participants.
disturbance, sorry.
Yeah, some participants.
No problem. You can go ahead.
Okay. So looking into the phenotype or the databases from the clinical side, so it was suspected that the subject is suffering, maybe having a familial hemophagocytic lymphohistiocytosis, so which is an immune disorder. So this familial, what you call as a HLL, is an autosomal recessive disorder. So which makes too many immune cells production will be there in these subjects, and these two T cells and macrophages that have been overproduced will infiltrate into various organs like liver, spleen, bone marrow, and also central nervous system, and resulting in a multi-system disorder. With the and this can be onset. It can be from a early onset. Some babies like from 2 to 4 years can be affected with this particular familial hemophagocytic lymphohistiocytosis. And there is a two forms of this HLH. That is, a primary form is a genetic predisposition, and the secondary form is due to a viral infections, which can be due to activated forms of the immune cells or the macrophages. So various studies have shown that the viral infections can elicit in some of the subjects who are having the genetic predisposition for the mutation in some of the genes responsible for the HLH. So that means HLH, the subjects who are having the genetic predisposition for HLH, in those subjects, if there is any viral infection, so that can trigger the development of the familial HLH. So when you look into the different subtypes of HLH, so you have 5 different subtypes of HLH. You can see from 1 to 5. So, and each one is there are 5 different genes that are responsible for different types of different subtype of HLH. So FHL1 is responsible type 1, and PRF1 is responsible for type 2, and so on. So now here from the clinical side, you have a data from the clinician that these are the clinical synopses that have been observed, and this is the suspected genetic disorder. So now we have the 5 different genes that have been already reported in the OMIM responsible for this HLH. So now in a genetic testing, we will target only screening on only these 5 genes. So what is the result of the genetic testing? Using this genetic testing will be performed using the next generation sequencing technology, using the targeted sequencing platforms. So the results showed that there was a mutation in a PRF1 gene in a region, exon region, that is a coding region 3 of PRF1. There was a mutation in a C2T. The change in the nucleotide was C2T, and the result outcome in the protein was arginine to tryptophan. So that means this change in C2T at the 673 position on the exon 3 of this gene was responsible for developing familial type 2. So it is an autosomal recessive type disorder. Since I already told it is an autosomal type of recessive disorder. So there will be both the alleles will be affected, or both the alleles, there will be mutation that exists in both the alleles of the chromosome, and hence it is called as a homozygous kind of mutation. And this mutation, that is 673 C2T, has been already reported in reported or seen in various patients, or scientific evidence are available to say that this particular mutation, that is C2T, is a pathogenic mutation, and it is having a high risk of developing the this HLH type 2. So hence, based on the scientific evidences and the online databases that are available, you can classify this mutation in this particular gene as a pathogenic gene or a pathogenic mutation. So now the clinician is sure that so the whatever the phenotype that has been observed in this patient is due to a genetic mutation that is occurred in a PRF1 gene. So now he has to decide what kind of treatment modalities has to be performed. So now the treatment modality that was decided was the bone marrow transplantation. So this patient underwent the bone marrow transplantation, and now the patient is doing good after 18 months of the treatment. So this is how, using the genetic testing, it helps the clinician to identify the cause of the genetic disorder, and also what kind of treatment he can decide not to cure the patient. So here the genetic testing is not only restricted for the immune disorders, it can be applicable for a various spectrum of disorders, including the, as I mentioned, cardiac disorders, renal disorders, ophthalmological, respiratory symptoms, neurological disorders. So whatever the clinical findings that we observed in the clinicians in the clinical database, so that can be identified using the genetic testings to identify the causative factor, whether it is due to a genetics factor or it is due to any other environmental issues that can be decided by using the genetic testing. So here you can, I am giving a one more example, like why how it can, this genetic testing can be implicated in a pharmacological studies. So here there is a boy, I mean, there's a patient who was getting treated for osteosarcoma. So, but here this patient was, when he was treated with methotrexate, he was suffering from an intolerance. So that means this methotrexate was not completely cleared from the body that was used for the treating the osteosarcoma. So when the clinician identified that there is an intolerance for this treatment, so they decided to identify what is the reason for this, and they went for the genetic testing, and they identified that there was a mutation in the polycystic kidney, PKD1 gene. So this PKD1 gene is responsible for polycystic kidney disease, that is in the autosomal dominant fashion. So that means any mutation in only one allele is responsible for developing this polycystic kidney disorder. So now, since there is a mutation in this polycystic kidney disorder, so now there will be abnormal functioning of the kidney. So this abnormal functioning of the kidney is responsible for that. Mean the drug that has been used for treating this osteosarcoma in this subject was not being cleared from the body, and hence there was an intolerance that was being showed. So now the doctors are clear now that this is because of the boy is already subject. I mean the subject is already having a genetic predisposition for the polycystic kidney disorder. So now the methotrexate treatment cannot be given, cannot be used at the regular dose what they are giving. Now they have to reduce the dosage. So now they will decide what the dosage has to be given to treat this, to treat this subject with the methotrexate. So this is how the genetic testing will help the clinician to decide even the dose response for the subjects that undergo for various chemotherapies. So and finally, so I would like to conclude with this slide, how this genetic testing can help. So the genetic testing can help you to diagnose the disease. Secondly, it can help you to identify the gene changes that are responsible for an already diagnosed disease. And thirdly, it can help you to determine the severity of a disease. That means how risk the patient is at for developing the disease. And also it guides the doctor to decide the treatment modalities for a particular disorder in an individual, and identify the gene changes that may increase the risk to develop the disease. And it also, genetic testing also helps in identifying the gene changes that could be passed on to the children. So it is, what is the risk of passing the mutation to the offspring can also be identified using the genetic testing. And lastly, it can also be used to screen the newborns for certain treatable conditions. So like how I mentioned, like phenylketonuria, so some of the other neurological disorders, so can be detected using the genetic testing. So that's how for this I will conclude my talk. So now the session is open for discussion. Thank you.
Dr. Puneeth Kumar, thank you very much for extensive view on giving the importance of clinical genetic testing in assessing the lot of disorders and the disease provision in the pre-birth as well as after birth and during the aging process. So now there are a few questions that has been dropped down in the chat box of Zoom and as well as in the comment box of the Facebook. I'm just going to ask you in behalf of them just to transact with the interaction. The first one is someone says, like, if the mother is positive to an oncogene or a cancer, oncogene, BRCA or any other thing, is it possible for particular organ? Is it possible for the child to get it, the mutated gene, mutated cancer gene in the during the birth or in the progeny formation?
That we have to decide. The first we have to look into the mutation type of mutation, actually, whether it is a heterozygous or a homozygous kind of mutation. So based on that, you can analyze the risk. Hello. So you can analyze the risk of being inheriting that mutation to the offsprings. So first we have to identify the what kind of mutation is there. So without identifying the mutations, it's like vaguely I can't tell you like this can be, oh, you have a certain tumor that can be inherited to your offspring. So it cannot be told vaguely like that. First we need to identify the mutation, and we need to see whether it has been scientifically, these mutations have been shown. So based on these identifications, we can analyze the risk factors of transmitting the mutation to the offsprings.
Yeah. The other question continuing to that is, if a mother got an adoptive gene mutations during the environmental exposure or the toxin exposure, right? Then he got a pregnant, and then is it possible for identifying that mutated gene in the neonatal stage or the growth
prenatal
stage?
Stage. Prenatal stage. It is possible to identify the risk factor of developing that, getting that mutation in the prenatal stage itself. That's how I mentioned in one of my slides that you can take the amniotic fluid or the chorionic villi sample to analyze the risk factor. You can identify the genetic mutation. First, let me take it in another way. Like first you take the mother sample, okay, mother blood, and identify the mutation, and take the amniotic fluid or the chorionic villi sample, and then identify the mutation, whether the same mutation is present in this sample. So based on that, you can identify the risk factor. So if the mutation is carried out, then you can say that of course the baby is in the risk of developing this genetic disorder. If the mutation is not there, so then you can say that the baby is safe, and he doesn't carry any of the mutation that is present in the that was found or identified in the mother. So that's how you can do.
Yeah, there are like a lot of questions. There were a lot of questions that has arisen with a single orientation of the point. Let's say like how we can decide whether the baby will have some kind of a genetic predisposing disorder. So how can we assess the risk, and to which category we have to ask them for clinical genetic testing? So what are the possible criteria that the clinicians will choose to decide this pregnancy has to go with the clinical genetic testing to assess the possibility of the genetic disorders?
Yeah. First they will check the family background. Okay. So if any of the family members, not only the circle relatives, even the second, third circle relatives are having any family history of this genetic disorders, they will check. And then the second point is they will check when you analyze the mother. Okay. So they do certain testings. Okay. So when they do the certain testing, if they find any clinical synopsis or the clinical data supporting to that particular genetic disorders means, so then you can have the baby screening. So that's how you will screen. These are the certain criteria that the clinician will consider to decide to go for a genetic testing or not. Most probably in the regularly in most of the clinicians, if you go, so they first, if a mother is a pregnant means, so they go for a karyotyping to check the Down syndrome, that is a chromosomal abnormality, whether chromosomal abnormalities are not are being there or not. First primary screening will be done using the karyotyping. But sometimes karyotyping cannot tell the entire sequencing mutation. So now the clinician will decide to go for the genetic testing based on the family history or any other backgrounds. And other factor the clinician will also consider is the what do you call that? So the age group. So that means if a woman is pregnant from pregnant for around 33 to 40 years of age means, or more than 33 or 35 years age means, then there is a chances of developing certain genetic disorder for Down syndrome or any other genetic syndrome. So then the clinician will directly refer for genetic testing. So based on the age group also they will consider, and if there is any, and there are also other factors that happens within the family members. So if there is a marriage that happens within the family member, then the doctor clinician can also recommend for going for a genetic disorder. So because we don't know, some of the family members may be a carrier for 5 to 6 generations, and it may all of a sudden it may appear in the next generation. So we don't know whether in which time the genetic mutations can be shown up or shown up as a phenotype. So in those cases, the clinician will decide to go for a genetic testings.
Yeah. So the other question is just extending that. We know about the genetic inheritance of the disorders, but is it any chance that during the growth period or during the aging period, there is a possibility of the genetic disorders that are arising, and what is the possible therapeutic for that? Can we able to cure? I mean, this is the question that they're asked. Can we able to cure this genetic disorder? Do you have any therapy?
No, for certain genetic disorders, there is a cure. For example, I mentioned here that by bone marrow transplantation, we could cure the HLH. Okay. So there are certain genetic mutations which have a later onset stage. I mean, so for example, the polycystic kidney disorder, which one I was mentioning in my last but one slide. This polycystic kidney disorder, the mutation in the PKD gene, sometimes what happens, it takes a late onset. So that means the patient will be, the subject will be normal till 25 to 30 years of age. Suddenly the mutation can onset at the age of 40, 45 years. Okay, so which can decline the kidney functionality. So in those cases, the treatment modality, it will change, or it will be decided by the clinician depending upon what kind of mutation has been occurred and which organ has been affected. So based on that, the clear treatment modalities will be decided.
Yeah.
So in case of if the kidney functioning, abnormal kidney functioning is there, then and it has already gone for a later stage where the kidney has completely stopped functioning. So then in that case, the doctor will go for transplantation. So that is the only option that you have.
Okay. So there are two questions related to the testing methods. The one asked, how can one rule out the differences across the results of a multiple test methods of clinical genetic identifications?
Pardon? I didn't get it. So can you repeat it?
How can we rule out the difference across the results of two different tests that has been conducted for the same kind of detections?
No. You cannot get two different kind of reports if you are doing a next generation sequencing. If you are doing a genetic testing using a next generation sequencing or even a Sanger sequencing means, so that means you will be targeting that particular gene sequence only. So the sequence, if you do it in two different lab, means it will not vary from one lab to other lab. So the sequence will remain same if you do in 100 labs. So the mutation that is present will be identified, and you will not get two discrepancies in the reports. So you will get the same. You have to get the same report. Otherwise, you can say that in any or the technology or the methods they are following is not the accurate method. It cannot be vary from one lab to other lab.
Yeah. So the other one, the last questions, I'm going to end the sessions after this question. How far the NGS, the next genome sequencing, and microarray will be helpful to detect the mutations in the virus? Or we are going to use this to detect for the COVID-19 because it's going to change from one area to another area? I think somebody is connecting to the COVID.
So here, let me point out that COVID is not related to any mutation. It's a viral infection. And these viral infections, already you know the sequence of this virus, and it can be detected by general real-time PCR methods only. So the sequence can be identified by real-time PCR method. And I don't think COVID has any implication on the mutation. But COVID might have a sequencing mutation from its earlier wild type to the present virus. That can be detected.
I think the question that they are stating is there is the variety of the viruses that has been identified are isolated are quite different from the native one because they have a slight chance of mutations and the RNA is continuously getting mutated from the origin from China to again Italy to India. They're asking, can we use NGS and the microRNA method to detect the variations in the genetic mutation that has been arised in this COVID-19 virus?
Yeah,
So
definitely you can detect using the NGS and microarray, but you need to establish a library for NGS next generation sequencing. You need to establish the library and also the database that has to be available. And the scientific evidences should be there. So it may take some time to build up these databases and libraries for identifying the sequence variation. But currently using PCR, simple PCR, you can identify the sequencing. So definitely you can use the NGS and microarray, but you have to build up the library and the flow chart for that, flow cell for that.
Yeah. Oh, thank you so much. Poornima madam, so if you have any questions, you can just ask and conclude the session. Poornima madam, please.
Okay, sir. Thank you, Dr. Puneeth Kumar, for your extensive view on genetic testing in autoimmune disorder. And you have explained in a very simplified way. And I'm sure that our participants have been benefited. Thank you very much, sir.
Thank you. Thank you for giving us this opportunity. I would thank you all for Mr. Dr. Pramod and also other colleagues who have given this opportunity for me. So thank you one and all.
Yeah. Thank you, Puneeth sir. We'll get back to you. Thank you so much. So we'll get a break for about 2 to 3 minutes for the next session. And the next session is going to be from Dr. Poornima Priyadarshini, and she's a scientist at CSIR-CFTRI. So we'll get back to the session in another 2 to 3 minutes. Poornima madam?
Yes, Pramod.
Are you? Oh, yeah. Okay.
Sir?
Hello,
Yes,
Poornima,
sir.
is it
Yes,
audible? Yeah, madam,
yes,
I think, yeah, I'm going to speak with the speaker just to confirm our voice is clear. Poornima?
Yeah, tell me, Pramod. Are you fine?
So is I'm audible?
Yes, you are.
Okay, thank you. So are you ready? So can we go ahead with the session?
I think this can, yeah.
Okay, yeah, so I can visualize you now.
Yes.
Okay, so welcome you all. So it's my pleasure to welcome for the second session of today's national webinar series. The second session, the speaker is Dr. Poornima Priyadarshini from CSIR-CFTRI Mysuru, and she's going to deliver a talk on food bioactive as immunomodulators during viral infections. Now, I request our faculty member, Dr. Puneeth Kumar, to introduce the speaker of the day. Poornima madam, please.
Okay, sir. Thank you, Pramod, sir. Good afternoon, everybody. I am so pleased to have the chance to introduce our guest speaker, Dr. Poornima Priyadarshini, CG, Senior Scientist, Department of Molecular Nutrition, CSIR, CFTRI, Mysuru. Dr. Poornima Priyadarshini did her BSc education in 2000 from Regional Institute of Education, Mysuru, and completed MSc Biotechnology in 2002 from University of Mysuru. Madam has completed her PhD Biochemistry in 2008 from Indian Institute of Science, Bangalore. Dr. Poornima worked as scientist in CSIR, CIMAP, Bangalore from 2012 to 2015, and she served as scientist in CSIR, CFTRI, Mysuru from 2015 to 2018. From 2018 till date, she is working as senior scientist in CSIR-CFTRI Mysuru. Awards or prizes or fellowships of Dr. Puneeth include BMBF postdoctoral fellowship from Ministry of Education and Research, Germany in 2008. And Madam has many projects under implementation that include understanding the cellular targets involved in bioactive peptide mediated protection of pre-diabetes or diabetes associated cardiomyopathy, around 46.73 lakhs funded by Serb Government of India. And another project, sustainable and cost-effective production of stable anthocyanin in Saccharomyces cerevisiae by synthetic biology approach, about 47.18 lakhs funded by BIRAC DBT Government of India. Madam has two house projects also that include effect of dietary modulators in key targets in diabetes. Participating agency is CSIR, and another is enhancing research capacity and initiating integrated MSc and PhD program in the area of nutrition biology in CFTRI Mysuru. Publications of Dr. Poornima Priyadarshini include 10 different research publications in reputed national or international journals, and nearly 12 abstract or paper presentations in various conferences and symposiums. Madam has professional affiliations that include Society of Biological Chemists Life Member, Indian Science Congress Association Life Member, Association of Food Scientists and Technologists India, AFSTI, International Aid Society, IAS. Madam has teaching experience also and worked as subject coordinator for biochemistry MSc Food Technology course and worked as instructor for molecular biology ACSIR PhD course, as instructor for genetic engineering ACSIR PhD course. Madam research area include lifestyle disorders, diabetes and obesity, neurodegenerative diseases, molecular virology, causes for food protection and detection. Now, 4 students are working under Madam for their PhD degree and 5 students work for their dissertation and 2 students are working as project assistants under her guidance. Now, Madam is talking on food bioactive as immunomodulators during viral infection. With this brief introduction, I request Dr. Poornima Priyadarshini to start her session. Over to Dr. Poornima Priyadarshini.
Okay, Poornima madam, I think you can go ahead and start with your session. Please share your screen.
I will.
Yeah, I can visualize your screen. Yes.
Can you see the slides?
Yeah, I can. I cannot see the slide. I can see your window, the folder. You can just click on the slides. I can see the webinar. Yeah, it's opening. It's taking some time, I think. Yeah, and just. Okay, Poornima madam, you can just click one more time on the PPT file.
Yeah.
It probably is not.
Can you see the slides now?
No, I can see the file. PPCC in the S drive.
No, I have shared already.
I can see your sharing, but in the sharing, I can see the folder that is PPCGH. So you can just minimize this and then I can see your computer screen. That the folder name is PPCG H drive.
Okay. Only that is.
Yeah, and then the first file is the Davangere webinar PP presentation.
Yeah, I have just.
So you can you can click that on.
I have clicked it on right now, and it's opening here, but you can't see, is it?
No.
Let me stop share and then do it again.
Okay. Yeah.
Share screen.
Open with.
Yes, I can see the same thing.
Just a minute.
I think you can minimize this folder so that I can see the screen.
Oh, okay, okay.
Yeah.
I think that is also possible. Wait.
Yeah, open with.
Can you see that now?
No, I can see only that folder. You can minimize this file, this one.
I have minimized that one.
But what can I see is only the folder, this PC PPCG.
Wait, let me close everything down.
You're sharing your screen, but in the screen I can see only the folders. So that you can minimize, probably the slide will come in the next background. The second one. So if you just minimize it, I'm not seeing you the desktop. Black's desktop also is not available.
Okay, now what can you see now?
I cannot see anything, so it's not shared.
Okay.
Yeah. Now it says Pramod GN has, Puneeth has started screen sharing.
I have done that.
Yeah, so now the same thing came. So we can see the file, PPCG.
It's open.
So you can do one thing. You can just open that and then give your desktop, and then you can click the PowerPoint, which is open. Yeah, it's opening now.
It's opening, right?
Yeah.
Yeah.
Oh, I cannot. Oh no.
It's opening now?
Now it become blank and then again it refreshed.
Sir, Pramod sir.
Yeah.
ಸರ್
Hello?
ಅದು ಬಾಟಮ್ ಅಲ್ಲಿ ಪವರ್ ಪಾಯಿಂಟ್ ಸಿಂಬಲ್ ಇರುತ್ತಲ್ಲ ಅದನ್ನ ಕ್ಲಿಕ್ ಮಾಡಿದ್ರೆ ಬರುತ್ತೆ ಅನ್ಸುತ್ತೆ ನೋಡಿ ಸರ್.
ಮಾಡಿದ್ದೀನಿ ಅದನ್ನ.
ಪವರ್ ಪಾಯಿಂಟ್ ಮೇಲೆ ಕ್ಲಿಕ್ ಮಾಡಿ ಮೇಡಮ್, ಅದು ಓಪನ್ ಆಗುತ್ತೆ.
ಪವರ್ ಪಾಯಿಂಟ್ ಇರುತ್ತಲ್ಲ ಅದನ್ನ ಕ್ಲಿಕ್ ಮಾಡಿ.
ಬಾಟಮ್ ಅಲ್ಲಿ ಇರುತ್ತಲ್ಲ ಮೇಡಮ್.
ಅಲ್ಲ ಮಾಡಿದ್ದೀನಿ ಅದು ಆಕ್ಚುಲಿ ಬರ್ತಾ ಇಲ್ಲ.
But we can see only the file. But we can see we are not able to see even your desktop. You can just close this. Just close this file.
Okay.
You can copy this slide to the desktop and then you can delete it later afterwards. You can just copy that slide to the desktop and open from the desktop.
ಅಲ್ಲ ಡೆಸ್ಕ್ಟಾಪ್ ಕಾಪಿ ಮಾಡೋಣ ಫಸ್ಟ್.
Yeah, now I can see your desktop. You can put it up. Dear participants, please excuse us for 2 minutes. We are going to fix it and we'll be back to the session. So please be patient. And copy ಮಾಡಿ ನಿಮ್ಮ desktop ಬಂತು. I can able to visualize your desktop.
Now can you see? I've opened it from the desktop now.
No, I mean it says like your desktop and copy. You can close it. You can open your desktop now. Just open the desktop. You minimize this.
Yeah.
I'm not able to see your desktop. I saw your desktop and then it went off. Now it says the file is open and then copy. Copy is highlighting.
Now what is happening?
Just it says copy and the copy is highlighting. I mean You click on the PowerPoint, the right button, and then you selected the copy button. You can close this completely. You can refresh.
Yeah.
So completely you can refresh and start sharing again from the desktop. You can start sharing from the desktop. Madam, I think you can unshare.
Yeah.
You can unshare and start sharing again. Yeah, you can start share again.
Let me share.
Yeah, now I can see you. I can see your picture.
See me, right?
Yeah.
We are going to the desktop now.
You can share.
Can you see the slides now?
No, I think it's not shared. You can just click on the share. It's not shared. I can see mine now. Yeah, it's sharing now. Yeah, I can see your desktop now.
Okay.
Yeah, now it's open. Yeah, I can see your slide.
Perfect.
Yeah, so now, yeah, it's clear.
Check.
Okay, madam, thank you. Sorry for the inconvenience that happened in sharing the slides. Now I request Poornima madam to start the presentation. Poornima, over to you.
Sure. Thank you, Dr. Poornima. It is like Poornima to Poornima. Thank you,
Yeah.
Dr. Poornima, for your kind introduction. And
Thank you, madam.
And I'm also sorry for this inconvenience that happened. And firstly, I would like to thank the organizers of the Davangere University for this meaningful initiative, and also for giving an opportunity to share some of my thoughts. And I would like to specially thank Dr. Pramod for his nice invitation and organizing it well. So this is all about, I would like to talk a little bit on food bioactives as immune modulators during viral infection. So food is basically because I'm from the Premier Food Institute, and the virus is because it's everything about virus in the world, all over the world now. So we would like to stick something about to balance in the absence of drugs and other things or the vaccine for particular virus. We have to focus on some alternatives, and that could be some of the food bioactives as immune modulators. So food, as you all know, doesn't need any introduction. We live on it and we need it 3 or 4 times a day, and we just have it irrespective of what is in that. But if you look at biochemically or chemical compositions of that, it is nothing but a mixture of macro and micro molecules that is used by the body to sustain, growth, repair the vital process that furnish the energy. So it's the most important thing. And if you look at the cartoon, it says like eat less and exercise for everything, or otherwise you have to go back and change the genetic code of your parents. And that is
Hello, Poornima. Sorry for interruptions. So I think the voice, yeah, I can hear you. The voice is little low. I mean, if it is little louder in the Zoom, it is okay. It's better. Little, yeah. I think you can just speak with the open microphone instead of the.
Okay, fine.
Yeah, now.
Can you hear me now?
Yeah, it's louder now.
Yeah. So what I was trying to say is food doesn't need an introduction because we live on it, and that is what it is most important for each and every one. So if you look at another angle of food, which we generally don't look into unless even I used to do it before coming to CFTRI, I should admit that. So it is a mixture of micro and macro molecules that is used in the body of an organism to sustain growth, repair, and all other vital processes, and most importantly, to furnish the energy. And if you look into this cartoon, it simplifies or it tells more about the importance of food. It just says eat less, exercise, or you have to go back and alter the genetic code of your parents. And that is very, very difficult. We all know that. We cannot alter it. So food is not only that gives nutrition, there are many bioactives in it, and that is nothing but by definition, if you want, these are the substances that are present in a natural constituents that provide health benefit apart from their nutritional value. And simply to say, they are the molecules that are biologically active. And these biologically active molecules could be bioactive peptides. The source could be the protein part of the food that we take, or the polyunsaturated fatty acids, or it is a polyphenols, mineral elements, or bioactive polysaccharides, and other things that can play a very important role. Not even they can play, they have shown and proven to play a very important role in many of the metabolic disorders, inflammation, and cancer, and you name it, and they have a role in it. Coming to the viral infection, virus. Now, virus also doesn't need an introduction. Maybe few months back, for the common people, virus we had to give an introduction, but even every kid knows what is virus. Everybody talks about corona, and corona is all the way. That is the thing I thought, even for this virus, unlike all famous thing, I would give a title or a subtitle. The viruses are simple yet highly complex. So viruses, basically, if you ask for information on viruses, they are non-cellular because it doesn't have any of the cellular organs in it, and they just have a genetic material and a protein coat into it. And they're very small, smaller than the bacteria, and they range somewhere between 20 to 300 nanometer. So you can't even say it as a microbe, though it comes under microbiology. Still, the question and the equations are still on in that. And these viruses lack cellular organelles, as I said, and they don't have any mitochondria ribosome, and they are active only inside the living organism. So other than that, they're just the inert molecule that can be worked anywhere or left anywhere out. But upon reaching their host, which are specific cells, they became highly active, and you know what happens after that. So if you look into general features and classification of these viruses, the main feature, they have a capsid. Capsid is nothing but that encapsulate the protein genetic code, and the nucleocapsid is both the capsid and the genetic material. And there are protective proteins surrounding this nucleocapsid. And some viruses will have envelope, some are non-enveloped. In case of corona, it has an envelope. And this capsid is enveloped with a phospholipid bilayer, and it can be derived both from, they derive it from the host cell without damaging it. They just bud off, and this could be either nuclear or cellular membrane. And nucleic acids could be RNA or DNA. So based on these general features, there is a classification. It could be DNA virus or an RNA virus. It could be enveloped or non-enveloped. And it could be a single-stranded DNA or a double-stranded DNA. Either way, it can be single-stranded RNA, and it could be positive or negative. So based on that, you have a different kind of viruses that exist in nature. And coronavirus, what we talk is a positive strand RNA. So coming to coronavirus or the COVID-19, what we already know in all our televisions all day around, is a disease, is a complex disease that is caused by a respiratory syndrome viruses. And one of the viruses, corona, that is a severe acute respiratory syndrome coronavirus. SARS-CoV-2 is responsible for the havoc that has created across the world right now. So if you look into the TEM image of the coronavirus and also the predicted potential image of this coronavirus, we know that, as I said, it has a capsid, it has a membrane because it comes under enveloped viruses, which is outside with the membrane proteins, which is also studded or decorated with spike glycoproteins, which have a very important role in attaching to the cells. And we have a nucleoprotein membrane, negative positive sense RNA. And this particular RNA is a non-segmented positive sense RNA with a genome length of somewhere around 29 kb base pairs. And you have a genome that encodes for both structural and non-structural proteins. When I say structural proteins, that means they are involved in making the assembly of a protein that forms a capsid. So as I said, spike, envelope, E protein and M protein, these are the one that takes over as a structural protein because they decide the structure of a particular virus. Whereas there are many non-structural proteins, including RNA-dependent RNA polymerase. This is very much necessary for the virus because once it gets infected and come into the cytoplasm, it has to replicate. So what it does, it comes along with its own RNA polymerase, replicate initially, and then use all the host cell machinery in order to perform, live, replicate, and also spread to the another cell. So if you look into generally the entire life cycle, it is simple. As I said, the spike protein is responsible for attachment because the receptor on the cells, that is ACE2 in case of SARS-CoV-2, is recognized by the spike. Entry happens to endophytosis. And then as I said, since it is a positive strand RNA, it acts like mRNA of a eukaryotic cell or a prokaryotic cell basically because it produces a polyproteins. It doesn't need anything like that. And since it comes under prokaryote, you don't have an introns obviously. So it is a polyprotein which gets produced and then get cleaved to give different proteins. These proteins later assemble and then bud off, as I said, along with the cellular membrane which forms as an envelope. So virus release and then spread happens and it continues its life cycle. An interesting virus which was dated back belongs to the same coronavirus group. For example, it is to say that there doesn't have one receptor, they have 2-3 receptors. In case of MERS-CoV, that is a Middle East Respiratory Syndrome coronavirus, which has a similarity with the SARS-CoV-2, is a receptor that is used as DPP4 or a CD26, which I'll come later to this point. But the entire other life cycle remains the same. So in case now we know that there are food bioactives and there is virus infection, and we need to connect these two with in order to see whether they can do immunomodulation or they can have any effect on our immunity. So if you look at immunity, that is nothing but it's the body's ability to resist and eliminate the potentially harmful foreign materials or abnormal cells which exist within. And the harmful materials can be many. It could be a virus, it could be a toxin, a carcinogen, a fungus, a bacteria, parasite, or germs. It could be anything. Because every day we come in contact with all these things n number of times and still we don't get infected often. That means there is a system, like a defense system, that controls everything. When I say there is a system, it is not just an organ. It's very complex system which includes skin, the lining of the mucous membrane, secretions that including tears, mucus, blood cells, bone marrow cells, liver, lymphatic system, and many more. And most of these tissues have a resident immune cells. And these are our first line of defense, like the border security force that is right now playing a very important role in securing India. So when you look into a typical immune response, as a cartoon says it is, the first barrier is the skin. So you have so many cells, the minute it says there is a parasite or a robber or a thief, in other words, if I would like to say, there is an army that is already ready to detect. And there are some cells who look into it and they say, yeah, it is indeed a foreign body, not our own self. So if you look into the immune response, the first that is immediately that act to any viral infection in particular is the innate immunity. So external defense, as I said, skin, mucus and secretions would take care. Initially skin is the barrier unless and until there is a damage or an opening through which this virus can enter. Or as you know, in corona, you've already known that it is through that's how we wear a mask, through the mucus or through the tears. We shouldn't touch our face because these are the route of entry. Here, even in this case, what happens, this mucus that is there when we sneeze out, that is taken care. We rule out most of the virus infection. And the tears also, where there are the lysozymes, it can act as an antiviral property, whereas the secretions such as a sweat has lot of antibacterial, antimicrobial peptides that also can act against it. Apart from that, when you go, the internal defense system have many other things like phagocytic cells. Phagocytic itself says that it engulfs, it eats off, and antimicrobial protein, many inflammatory response, and natural killer cells which engulf and kill the complete cells. These are the part of innate immunity. When this is finally connected to the humoral response or the antibody production or the T cell mediated response, these are the acquired, which is lower, which comes little later. So this comes later. So initially the first few hours, that is first 6 hours of the infection, is because of the infection is taken care right away at our immune cells. So once the acquired immunity comes into picture, when we have a second infection, these are immediately recognized, and then the responses posed on them. As I said, 0 to 6 hours to 12 hours, this is the innate immunity, the first line of defense. If you can compare, I would say this is like a border security force. They're right at the border protecting us. Once they have performed their job, when there is some other invaders which pass through the skins, then the adaptive immunity takes place, like in our Navy or an Air Force or whatever. They come into picture in the form of B lymphocytes, T lymphocytes, or the effector T cells, and they take over, and there is a complete destruction of the virus infection. So as I said here, they work in different way. First thing is the protection against the infection. When the virus comes, the interferons or the cytokines are produced, and they are recognized by the target cells and then eradicated. Or in case of adaptive immunity, the B cell, the antibodies are produced which coagulate with the virus, which would precipitate and also kill the cell using the T cells. But then the question is, if so much of immunity is there, and if every one of us have our immune system working such strongly, then why do we end up with lot of infections? And why, at least in the current context, why so much of a havoc that has been created due to COVID? There could be many reasons, because viruses, as I said, they're simple, complex. I should also add they're very smart. So they have a different way of dealing with this immune system. So this slide tells in brief how this virus invade the total human system. So when I was listening to the earlier speaker, there was a question where they asked, this COVID is coming up with lot of mutations, if I'm right, and it is not same as that of which is originated in Wuhan. So we have lot of, can it be detected through NGS? That was a very good question. But in the similar context, what I would like to say is these viruses undergo mutations, one of the way to invade the immune system, so that the receptors can be recognized, but the cell is no more detecting it as a original virus particle which has already come into picture. So there is totally a difference in between the mutated one as well as the current infection. So the cell cannot mount the response that is supposed to respond for a virus. So this is one way of doing it, and this is from the outside. Or these viruses also make certain changes in their decoration, the alteration in the HA or in the spike protein, so that they like hide from the immune surveillance and then enter into system. Once they enter it, they completely hijack the immune system. And especially when the person is already having any other complications such as diabetes or a cardiovascular disease or a hypertension, they're already immunocompromised. Immunocompromised, when I say this entire immune system, both the innate and the acquired is not working at to the point which it is supposed to work. In that case, these viruses take over and they work in alter all the signaling pathway, and they also create a havoc, which is also, you would have heard the term called cytokine storm, wherein the immunocompromised people, the inflammatory markers, cytokines, which have to form a protective effect, is overexpressed so much that it is just attacking the other organs and damaging all other possible organs like lung, heart, kidney, and everything. So we have to be careful when we say the immunomodulation. It shouldn't suppress the active form of the interference, and also it shouldn't activate so much that we end up in creating another problem. The slide is so much. Not moving.
Hello?
So if you look into the other modification that can do, it can ubiquitinate, and it can also pose a TLR receptor, toll-like receptors, and those all that points it can be modified so that it can skip the immune system both in the innate as well as the adaptive immunity and go on with the next level of infection so that they can spread easily. That's what I wanted to say. Why is the slide not moving?
So you can just go back to escape.
Yeah.
And then you can just click on again. Maybe it has hanged, system has hanged. Yeah, it's moved.
But each slide, that's the problem. Okay. So, so immunomodulation, as I said, this is how the virus invade the system. And the concept of immunomodulation is not new to us. It dates back to centuries. That is in 1796, when Edward Jenner first gave the concept of vaccination without even knowing it could bring such a drastic change in the world of immunology. So what he just is, it was everything based on his observation. As you have already heard on your microbiology and immunology subjects, it is like he found somebody not getting infected where others were getting infected with cowpox and the smallpox. So what he thought, due to his observation, there is something that is coming out from milking the cow, or the cow that's been handling, and that is getting transferred, which is giving immunity. So the simple experiment, he just took the infected milkmaid cow paw and then the pus, and then co-inoculated to the others, and found that they were completely cured. That is how there was a first evidence which came there can be some substances outside the body that can modulate the immune response. So the immunomodulator can be a substance, a biological or a synthetic, and which can do both suppress or enhance any component of the immune system, including both innate or the adaptive arms. So this is called an immunomodulator. There is a problem with this. Can you see the slides, Pramod? No. Change. It's getting hung. Hello? Hello? Pramod, can you hear me? Hello? Dr. Poornima, can you hear me?
Hello?
Hello, can you hear me?
I can hear you. Your voice is clear. The slide has changed.
Okay, so I thought I was not heard. Okay, so as I said, there are many nutraceuticals or the bioactives from various sources. They have proven a very important effect on all the lifestyle disorders, including cancer, asthma, and also infections and so on. So if you come to some of the examples, for example, turmeric or the curcumin is the main component, beta carotene, it's present in carrot and other things. EGCG, which is a part of a tea, and many other components are known to have a bioimmunomodulatory compounds. That means they have an anti-inflammation properties. What do you mean by anti-inflammation properties? As I said, when there is inflammation, when there is a cytochrome storm, or when there is an overexpression of cytochrome, they keep attacking the other tissues and damaging. So there is an inflammation that is occurring. So that time we have to reduce their inflammation. In that context, all these bioactive molecules act as a anti-inflammatory properties, or else they also known to activate lymphocytes and other NK cells, so that upon infection, when there is an infection, that the number of NK cells and lymphocytes are more, so that they get activated to act against the invader. In our case, it's the virus. So if you come to the mechanism, it could be many ways. They might produce some of the interleukins like IL-10 and EF2, which is an anti-inflammatory marker, or it can be inhibit the pro-inflammatory markers. As I said, they can enhance and polarize the macrophages, so that the macrophages come to the place where is the first line of defense is happening, so that they can act there only and kill all the virus that is in, or the cells that are infected with the virus. Or it can either enhance, as I said, this is a thick mark, or it can also down regulate the pro-inflammatory markers and other stuff, so that it can bring about the effect that need to. So our lab in this context is working on many bioactives, and one of the important enzyme of target that my lab is currently working is a peptidase 4, otherwise called DPP4, in terms of diabetes and inflammation. Even you know that there is a strong connection between the inflammation and diabetes. So what dipeptidyl peptidase as such do, it has a two functions. One is a catalytic function, the other one is a non-catalytic function. In case of catalytic function, it does an enzymatic activity. I'll come to this later, where its substrates are GLP-1 and GIP-1. In the non-catalytic function, it is expresses over all the NK cells and other cells, and does the very important role in inflammation. And during lifestyle disorders, be it is diabetes or obesity or a cardiovascular disease, this particular enzyme is known to overexpress and creating lot of inflammation problems. So dipeptidyl peptidase 4, as I said, it is a transmembrane glycoprotein serine protease. That means it is a peptidase, basically cleaves off and has a serine at its active site. And it has an increased enzyme activity during diabetes and other metabolic disorders. And they have two incretin hormones, that is GIP-1 and GLP-1. This is basically an enzyme structure. And what is the exact role? When you this particular enzyme comes into picture after you have the food, which is called postprandial glucose homeostasis. So when you go to a blood checkup, they check both your FBS and PPBS. So this FBS is the one which takes over the initial fasting glucose level, whereas the PPBS, that is after you take the food, what is your glucose level is monitored. And in this juncture, the DPP4 has a very important role. So as it is shown in this cartoon, when you have food, there is an incretin effect. As I said, the GIP-1 and GLP-1, they act on pancreas, and pancreas in turn act on two-way, that is beta cells and alpha cells. They do the insulin secretion as well as a glucagon secretion, and there they act on both liver and fat cells, where there is increased glucose uptake and the liver decreased glucose production. So in this mechanism, they maintain the glucose level. And this particular incretin is a target for DPP4 enzyme. Both these are present in the intestine. So what happens during diabetes? These enzymes are overexpressed to so much fold higher, so that they cleave off all the incretins, that is GIP-1 and GLP-1. So there is a diminished effect on the pancreas. So there is no proper insulin secretion, neither glucagon secretion, and which will end up in hyperglycemia. That is too much of a sugar in your blood when you go for testing. So what we are trying to do is, can we come up with some bioactives that can inhibit this enzyme, so that we can gain back the glucose homeostasis. So there are some inhibitors already present. When you go to a shop, they would prescribe. These are called gliptins. But of course, all these drug molecules come with side effects. So we are looking into finding out some of the bioactive peptides. So one of my PhD student, Mr. Abdul, is working on generation of bioactive peptide fractions. It is his work where we are concentrating on various millets, because millets also known to have an effect on other lifestyle disorders. So we have right now generated the peptides, and their activity has been detected or proven through both the in vitro assays, where we do the in vitro enzyme studies, and also validated cells, the cell lines using different. In this case, since the site of DPP4 and GLP-1 action is intestine, we take a cacotucins, that is the intestinal cell lines, and we study their effect both in vitro and in cells. So once we have this millet fraction, we have further went ahead, characterized using various mass spec and other biophysical techniques, and found that we have come up with three important peptides that have a very good role in inhibiting DPP4. And we have also gone ahead to find through molecular docking studies that where exactly these peptides bind and how exactly they do the role. With this, we know that there is one of peptide 4, it is interacting right at the active site pocket along with the serine residue, and it could be a very important bioactive peptide target. So this docking studies, and now we are trying to see their interaction and how exactly work, so that we delineate a molecular mechanism of these peptides, so that later on if you want to give it as a supplement, or we can give it as a drug that can replace, or if you cannot replace the actual drug, at least we can lower the concentration of or, or the current drug, so that the side effect is reduced. That is one of the approach we are trying to do. And as I said, since this DPP4 is involved in inflammation, and inflammation is one of the major risk factor during diabetes, the target action also can be there through these peptides. Apart from that, here I would like to mention, apart from the symposium and Pramod, I have another Pramod with me, a PhD student from your Davangere University, alumni of your university, who is working on, that is a different connection. He's working on another protein which is called DPP10, dipeptidyl peptidase 10, and it is involved in tau aggregation, that is in Alzheimer's and inflammation. When I say that we are concentrating, when you say Alzheimer's, there are two things that comes into picture. One is the amyloid beta and also the tau protein, and we are concentrating on the tau aggregation, and we would like to have a molecules or a bioactives that can inhibit tau aggregation. So that's the kind of his PhD work, and he's working on it. Towards his study, he has already made a clovoid nanoparticle. Why nanoparticle? That comes into, that the entire question comes into a different area, because you may ask, now we take food every day, especially Indians, we eat lot of turmeric, we have lot of spices, everything is there in our menu, but still we have all the problems. India is going to be diabetes capital, we have lot of infection, COVID is seeing no control, and many things. Then you say, where is these bioactives are going? What is happening with us? So what we can answer in this thing, though the bioactives are there in the food matrix, how much it gets absorbed or how much bioavailability of these bioactives are there depends on the individual, and it also depends on various other factors. So nano encapsulation is one of the method in order to enhance the bioavailability and also bioaccessibility. Especially in case of Pramod's study, he has to target brain, where we have a huge problem of blood-brain barrier. So we have nano encapsulated this particles, that is the clavoil, and its active component eugenol. And initial studies have shown they are non-toxic at certain concentration compared to other controls, and also it is non-hemolytic, so that we can use it in therapeutic purpose. It also went ahead to shown that the clavoil inhibits tau aggregation in vitro, and we have a promising result show that it could be one of the therapeutics for Alzheimer's. Here is the TEM analysis with various concentration of our nano-encapsulated cloud oil, which degrades the entangled tau. And this is the in vitro assay, which shows higher the concentration of thioflavin, then you can know that the aggregation inhibition is positive. Apart from that, another student of mine is working on milk bioactives, especially she's concentrating on both GAGs and also milk peptides, and trying to say their inhibition towards obesity. So these are some of the work that is happening in my lab to prove the potential of these bioactives from different sources to get an access to, or to give a different perspective of looking into food as such towards the lifestyle disorders and inflammation. So at the end, I would like to acknowledge all my students from whom this work has been done, and also my collaborators, my funding agencies, and also Davangere University, the entire family of Davangere University for giving me this opportunity. Thank you. If there are any questions, I'll be happy to take it.
Yeah. Thank you, Poornima. Thanks for the extensive view, and you had given an overview of the infection of corona, the COVID-19, and how the mechanism of the infection, the pro-inflammatory mechanism, how it is going to increase its symptomability in alveolar tissues, and as well as the possible the immune protections that we have through interferon alpha and beta. So there is an extensive view and a lot of basic information, and you're also focused on the peptides, which can be an anti-inflammatory targets, which can take care of antiviral mechanism to check the growth and proliferations of COVID-19. And your extensive work has provided a lot of information. So we have some questions. There are a lot of questions because since it is dietary related work. And the first question they asked is, as you are from CFTRI, so from the general articles, they might have studied, and there are 6 or 7 questions related to the same issue. That what about the spirulina supplement that has been added? Even the ICMR and the AIMS are using it, which is the basic product for immune boosting and from the CFTRI. So how it is going to act and how it is helpful to increase the immunity during COVID surveillance?
Actually, what has happened, we have spirulina work has been done very extensively in CFTRI, you have known that. Now we have incorporated the spirulina in various food matrices that can be taken, and it is part of many of the school meals and other things we have tried out, and we have shown that it has a better effect. The immunomodulatory effect, which is known right now for spirulina, maybe for various reasons. One is it is a rich source of vitamin C, which is an antioxidant. So that itself takes care of the many of the initiation of the inflammation. Second one is the phycocyanin that is present in the spirulina, which is known to increase or inhibit the inflammation that occurs. So it downregulates the inflammation pathway. So it's basically what happens. Why I think there are many studies need to be done in order to claim that is an immunomodulator. Of course, it is known for other normal patients, especially in case of COVID. I would say since there is a lot of cytochrome that is rushing in the COVID patients or with the immunocompromised patients, if you suppress some of its effect, there itself some of the modulation is taking care. So that is the kind of effect that is seeing. That's why I think it is working with the COVID patients as an immunomodulator. So still many studies need to be done because it has a ample of other bioactives. We don't even know whether there is any molecule that can inhibit directly COVID, or it can target any of the life cycles. We have no idea on that as such now. And spirulina has highest protein content in it. So even the protein part and the peptides that are generated from the spirulina, how they are acting, that is still yet to be understood. But as of now, what we know is because of the vitamin C and also phycocyanin that could be acting as an immunomodulator.
Yeah. So other question is, it's a basic questions. I think some students had asked it. They just want you to elaborate on the coronavirus infection and what is the action on the respiratory system, and how serious it will become when when it enters into circulatory system, into the blood.
Actually, we
Multiple
know.
organ infections. Why it does the multiple organ infection?
That is what it is. The answer is there in the question itself. As I said, coronavirus, the current COVID infecting, that is SARS-CoV-2, what we are talking here, that is originated from Wuhan, we actually doesn't know still the origin of it, and it is totally different from what has been reported earlier SARS virus. So it is more lethal. What is happening? It is directly attacking the alveolar cells in the lungs. Lungs are like, it is one of the organ you cannot reach also, like that of the brain. And in the absence of any vaccines or any medications, we are not knowing how to handle this particular virus. That is the main reason. That is a big problem that everybody is facing. We know, like, there are drugs that are getting developed where we can stop the virus infection, right? If you remember the life cycle, the first step in the life cycle of virus is to get an access to the cell. That is the attachment, true attachment. It has to get attached through a receptor and then enter into cells. So many people are trying to identify certain molecules that can inhibit right there, or they're trying to coming up with an antibodies that can inhibit this particular attachment process, so that right there you can stop it and you don't have to go inside to work on it more. Of course, the other molecules that are in, I mean, they are repurposed right now or on protease inhibitors or the replicase inhibitors. So these are worked in the other viruses. So they are thinking they can work it here. But you can see from the news and other factors, they're not going that way. That is the reason, in the absence of any medications, we are having the huge problem. But there again, I say there could be reason, like the kind of spice and other things that we take, and also minimum measures they're asking us to drink, take a lot of amla and all those things. They are helping out to some extent to keep us good enough to face it. I don't say that we are just virus free, or virus cannot infect anybody, it can do it. Only thing is that I can only say that from being uh, having a good immune system or a boosters with us, we can see that we can defend better. That is the only thing I can say.
Yeah. So the other question is, do SARS-2 COVID has a lipid bilateral membrane and it has been known to have it? What is the advantage it has compared to other similar viruses?
Yeah, it does has, because it is chipping off from the cell membrane, and if you see the structure of it, it has a bilayer and also all the spike proteins are attached to that. So enveloped virus are a little difficult to handle other than the non-enveloped. So in the case of non-enveloped virus, the access is easier. Whereas here in the lipid bilayer, what they have, the access is difficult and their genome is very well protected within. So that itself acts as an advantage. And they can reposition their spike protein so well, so that they can also modify so much that they can just get through. That is an advantage. The other equally disadvantage is that it has a bilayer and we have a membrane which is similar to that. The phagocytosis is also easier. It can directly enter endocytosis is easier. It can directly enter the cell easily, comparable to other viruses.
Okay. So the other question is related to your research presentation that you had done. Has the molecular dynamic stimulation of the peptide and DPPH has been done? If so, how stable is the interaction?
Actually, we have done the molecular docking studies, and it shows it binds into the binding pocket. That's how we came to know that they are interacting very specifically. Apart from that, to see how stable they are, we have done the other studies, binding studies, and the interaction is somewhere in the micromolar range. So I personally feel they have a very strong interaction with each other. It's unlike almost like other drugs that are available.
Yeah.
Yeah.
The other question that the other question that they have is, there is a threat for the oxygen during the COVID infection. Is it because of alveolar tissue damage, or is because of the COVID virus to change the structure of the heme? So there is two philosophies, and someone asked, like, Is the oxygen deficiency is created by the virus is because of the destruction of the alveolar tissues or because of its interactions with the change in the binding specificity of the heme in the blood? That
See,
is the previous question. The same person had
yeah.
asked this question.
Very frankly to say, I should say I don't know the exact answer to that, but I would presume it's both. Once the alveolar cells are damaged, once the lung cells are damaged, the access of the oxygen that you get and the hemoglobin that circulates both have an effect. They are interdependent. So what I feel is that it is the both. That is how not only lung is getting infected, the virus that pass through the blood circulation and enter into other cells, other organs also are equally getting damaged. I presume that it is the both. But very frankly, I would say I have to look into it.
Yeah. So the other is a very generalized questions again on spirulina. That some people they are asked, can you suggest who all has to consume spirulina and its product? When we need to consume, is it essential uh, a prophylactic or therapeutic?
See, please, I would like to suggest, please consider as a supplement, dietary supplement, not either as a therapeutic or as a prophylactic, because spirulina is such that you won't get anywhere so much of a protein and other all minerals that spirulina has, so that you can have it anytime. Only thing is that spirulina has a little off smell, so that people don't like to have it. So that is being modified. There is a technology in CFTRI is developed to mask this odor and other effects. That's how it has been incorporated in chickpea and other things. That can be taken for sure. Apart from that, I would also suggest, because the spirulina has been, if in Ballari, if I remember, the Jindal has taken up one of the program to give spirulina supplement to the school and Anganwadi people, and the primary school students and all. They have done the survey, and it is shown very promising, even with their height, weight, and other measurements they have done, and it looks very promising. So I suggest it can be taken as a diet supplement instead of thinking in a therapeutic view.
Yeah. So then the last question, like somebody asked about the wheat and the immune arthritis and the the gluten intolerance, and they are connecting with the gluten and the autoimmune disorders. So they just asked about your opinion whether the gluten induces autoimmune disorder. It's
Yeah,
probably out of the content,
yeah.
but still.
No. I was also thinking about it. See, there are many gluten-free products that are coming up. So in that context, and also there is a now FSSC, that is a Food Safety and Quality Control, has made it mandatory to see that the gluten had to be detected in
Yeah.
sample. So many people are prone to gluten related whatever the other problems,
Yeah.
inflammatory problems.
Please, I
So
probably if I want to add, maybe if the gluten is intolerant, the bacterials are going to produce certain
yes.
gluten hydrolyzed peptides, and these peptides can mimic the endogenous antigens and probably may initiate immune responses, which can induce autoimmune disorders. That's the only thing I can give for the
Could
audience who
be,
had
because I'm not worked in that angle, so I wouldn't be able to comment so
yeah, I
strongly.
think it's entirely a different one. So
Yeah.
so there is last, yeah, I think we addressed this question. I just asked, I just saw the comment and asked that. Okay, so the question that came just now, so the same thing. And finally, the question is, there is a lot of hydrolyzed peptides that are available as a supplement, nutritional supplements. So
Yes.
is the whole protein is beneficial as a protein source, or the peptide supplements, or the hydrolyzed peptides? Which one you want to recommend as a high protein, nutritious, immunomodulatory substance
I
to
would
recommend for
say,
health benefits?
I would say when you take a whole protein as such, it has to go through your digestive system, right? So it has to go through, and the peptide that is released are different and different kind, and they have its own effect. When you say protein hydrolysates, they're already processed. And if some people have a problem in their digestion, especially the elderly who are compromised with all other this thing along with their digestion problems also, for them, these hydrolyzed will have an edge over others. And apart from that, these hydrolyzed will further go degradation, and then they will end up in proper response. So that what we have done in our case, in order to produce our peptide, we have mimicked the same thing that happens in the intestine. So we use different enzymes that is present in the gastrointestinal tract, different pH level, digest, and then we see the effect, so that we have almost mimicked what happens within the system. So that is how I feel that could be used both.
Yeah. Uh, Purnima, madam, thank you so much. It was an extensive view, and you had answered most of the queries that was present. Now, I request our faculty, Dr. Puneeth Kumar, to give a concluding remarks. Purnima, madam, please.
Just one thing I would like to add. It's like as a famous says "Utta ballavaniga rogavilla." So we should know what we eat, and because it says, as a cartoon I put, it says that 100 years of medical progress, and what you all suggest is eat less, eat what you want. So we shouldn't wait for somebody else to say. We should act on now and eat healthy and be healthy. Thank you so much.
Thank you so much. Purnima, madam, please.
Okay, okay, sir. Thank you, Dr. Poornima Priyadarshini, for your excellent and extensive view on food bioactive as immunomodulator during viral infection. The topic is very much related to the present situation, COVID-19 situation, and it has educated many of us to understand how food itself is acting as medicine during viral infection. Thank you very much, madam.
Thank you. Thank you.
Purnima, madam, thank you so much.
Okay.
I will thank on behalf of Department of Biochemistry, Davangere University, and as well as on behalf of Society of Biological Chemists, Davangere chapters for giving you for sparing your valuable time and giving an extensive view in your presentations. Thank you so much. With this, we are going to end these sessions. Thanks a lot, madam.
Thank you. Thank you for the opportunity.
With this, we are concluding today's webinar session. Thank you all. Good day.